is Jeff Welser from IBM Research, and he'll be talking about the microbiome of food.
So hello. Not to malign any of your hygiene techniques, but let's just acknowledge we are all covered with bacteria. In fact, the average adult has about 40 trillion bacteria cells on them. If you consider that the average adult has 30 trillion human cells, you can easily see we're outnumbered, even on our own bodies, which to some of our earlier conversations might make you wonder who's actually in charge. But leaving that philosophical discussion aside, clearly it's a very important part of our health ecosystem. And of course, bacteria covers the food we eat, the plants and the animals it comes from, the soil it's grown in. So most of that bacteria must be benign. And in fact, we're finding increasingly it's incredibly important to our health and well-being. But there are a few bad actors. 1 in 6 Americans this year will suffer a foodborne illness, hopefully no one here tonight. And about 2 million people actually die still around the world from foodborne illness. And it's the usual folks, E. coli, Salmonella, Listeria that we test for. In fact, the food industry spends billions each year testing for everything they know that's out there. But increasingly, it's difficult. The food supply chain is very global. You can't control all parts of it, and there's lots of different places things can slip in. So they've been looking for new ways to do testing that would give them headlights to the things they don't know they're supposed to be looking for. And this enters metagenomics and IBM. So metagenomics is a way of using genetic information to classify the microbiome, the bacteria that's on anything. So let's say you take a sample of chicken or some wheat that's going into a food processing plant, you put that into a next generation sequencing tool, you slurry it all up, and you literally sequence every bit of RNA and DNA you can find in it. Now, you aren't going to get anybody's full genome that way, but you will find out if you take that and match it against a giant database of all the known genomes of bacteria and viruses and fungus and mold, what is the microbiome? What's every microbe and the relative populations living in that sample? And if you do that every time you get a shipment in, you'll get to see a baseline. This is what the healthy chicken or wheat sample looks like. Now, if you ever see E. coli or Listeria, you'd, of course, not use it. But what if one day you just test it and you find that there are certain bacteria you haven't seen before that are there, or other bacteria you're used to seeing are gone? So there's been a shift in the local microbiome ecology. That gives you the headlights that something has changed here. And this is a big data problem. You're talking terabytes of information for each of these and doing this giant matching in the database, which is why IBM got very interested in it. But it gives you a way then to start to apply analytics and AI and machine learning to understand what that change means. Now, maybe it just means it's the rainy season, so you'll learn what the seasonal changes are. But maybe it means there's some unknown pathogen that's in it, or maybe something that's not even a pathogen. Maybe there's extra heavy metal or some plastic that got into the slurry. That tells you something's changed because the microbiome itself is acting as a microscope, testing what's in its local environment there. So we're working on that, and at the same time you learn all sorts of other things, like you'll find out if that $24 a pound fresh halibut you're buying is really halibut or maybe tilapia. The genomes obviously are very different. But more subtly, as we start to use AI to study the patterns, we hope to also be able to tell you whether it was actually fresh or frozen, because presumably that process will actually change the microbiome. Or when you buy olive oil, it will tell you if it's actually olives or maybe a little corn oil that's been slipped in there. But even if it is olives, hopefully you could also start to learn, if you study it, whether those olives actually came from California or Italy. Might be the same species of olives, but you can trace what the microbiome is telling you it's coming from. We also hope more speculatively to learn, are certain microbiomes more conducive to preserving food longer? So this could help with the spoilage and issues that we have in the food supply chain today. About 30 to 40% of the food that we produce never goes anywhere because it spoils somewhere along the process. Not to mention the bananas sitting on my counter, which seem to go brown in like 2 days. So if we could figure out ways that microbiomes could preserve that, that would be a wonderful way of also improving sustainability. So IBM's interested, obviously, in all the data behind it, but we've partnered with food companies like the Mars Corporation, testing companies like Biorad, and universities like UC Davis, Cornell, and just recently UC San Diego, because there's a lot of science behind understanding what this really means. And in fact, at San Diego, they're actually studying the human microbiome to understand the effects it has on health, specifically for dementia and aging-related diseases. So this kind of brings us all full circle now. So if we are what we eat, and if you'll let me use the royal we to mean my body plus all the microbes that are with me on the journey, I'm feeding all of them. So if, in fact, I can find the microbiome of the food, it might make my food supply chain safer and more sustainable. It might also eventually lead to longer and healthier lives for ourselves. Thanks very much.